Hi everyone. My name is Rhema. I work I'm a PhD student at the American Museum of Natural History in New York City. Um, and today I'm just going to be talking about something I've been working on for some months now, which is trying to evaluate the efficacy of using eDNA as a tool for coordinate diversity assessment in tropical regions. Um, So first of all, just before we delve into everything, a little about eDNA itself. So as dragonflies and organisms move around in the environment, they leave fragments of DNA in form of skin cells, feces, sperm, and different materials in the organisms and in the environment. Sorry, and these form fragments of DNA of the organisms and leave traces of these organisms in the environment. And this deposited DNA can be extracted from a water sample or a soil sample, and is what we refer to as, eDNA. And in theory, rapid diffusion of DNA means this DNA of our target organisms, in this case, dragonflies, can be detected anywhere within the water body and not just at the point of its origin. And so this technique allows us to detect our target organisms without need for physical capture of the organism, which we know can sometimes be indiscriminate, especially when we're working with rare and endangered species. So this tool has been proven to be essential for biodiversity assessments, especially, like I said, considering cases where these target organisms are endangered, some of them are elusive, transient, and just generally difficult to detect by traditional sampling. And so previous studies have shown that eDNA, this technique, has been more sensitive in the sense that it was found to detect target organisms in areas where they couldn't be detected by traditional sampling. And this photo down here just shows a simplistic overview of how the whole technique works. Because when I was first interested in this technique, I came across this photo, and that's why I put it here. And so it just gave me a brief overview of what it entails, although it's, I will tell you, generally more complicated than this for sure. So you just get your water samples, water sample gets filtered, the so the membrane that traps all the DNA, and then we can use it for some downstream analysis for metabolic coding or just analyze to detect single species. And although this is a really innovative technique, there's still a lot of things that are yet to be understood about how this technique works and the success of these techniques, and that leads to some of the knowledge gaps in this field, which I'm interested in. And so a lot of the previous studies have been heavily focused on aquatic vertebrates, mostly fish, and a recent study was able to prove the positive correlation between target organism biomass and the success of this technique. So generally using this technique for aquatic invertebrates, which are relatively smaller in size and have a relatively smaller biomass, has not fully been explored. Additionally, previous literature have been heavily focused in regions in North America and particularly in the US and Canada. And so there's a huge, I've seen there's a huge variation in, environmental, excuse me, environmental and climatic conditions between biomes in, temperate regions here in North America and tropical regions in, South America and Africa, for instance. So there's a lot that needs to be, uncovered as to how this technique might, hold up in these areas. And so the aim of this study was, to use dragonfly eDNA for a preliminary and baseline study, to evaluate the success of this technique when being employed in the tropical regions, especially in the study of aquatic invertebrates, and particularly, dragonfly in this case. And so we hypothesized that, due to like I said, the environmental variations between these two regions, these two might not, be as efficient in these other areas, and we have some shortcomings, and we'll have to make some tweaks here and there to get it to work optimally. So for the sampling, We looked at regions. We looked at regions in Nigeria, which I just highlighted here on the map, and just honing in, going to the methods. The water samples I collected, I filtered using filter towers and stored them in 100% ethanol to be taken back into the lab for the DNA extraction using bashing beads. And so I took 2 replicates from 3 regions of the lake. So I divided the lake into 3 regions: a lower region, a middle region, and an upper region, just to maximize representation. And so I collected 2 replicates from each of these regions and just ran the whole process of filtration and the DNA extraction. And so we used bashing beads to help with the lysis of microbes and some of the other contaminants we might have picked up. And so this just shows a workflow of the methods that were involved. We had to run a lot of these methods over and over again, running a lot of PCR, optimizing protocols. And so as we moved on, we kept on encountering a lot of non-target DNA, because it's basically a soup of DNA where we have microbes, we have, plants, we have fish, we have whatnot in the water samples. So we needed different iterations and optimization processes to get the desired results. And so one of these steps was a further cleanup step using magnetic beads to help us read the PCR products of any contaminants that might have, slipped through the cracks in the DNA extraction process. And so despite trying to get rid of all the off-target amplification and the non-target organisms, which is the main challenge, I would say, for this technique, we still had to carry out some gel extraction, gel electrophoresis, where we used blue light. We just put the gel under blue light, and then we just cut out the right amplicons we were looking for, which was exactly 450 base pairs. And so we just ran those through some reagents and with that, we're able to get the particular extract of the target organism of the dragonflies we were looking for. And yeah, like I said, it's a soup of DNA of different organisms. So we just ran a metabarcoding for parallel sequencing of all the taxa present in the samples. And we carried out some library prep, which is just essentially pulling together different DNA fragments containing adapter sequences that are compatible with the sequencing method we wanted to use, which was Oxford Nanopore. We did that for each individual taxon present. And so for the bioinformatics and downstream analysis, we just used AliView to align the sequences. We used Mesquite we had a lot of insertions and deletions and gaps in our sequences, which we understand because we weren't using, it wasn't great DNA quality to start with, because this DNA has been acted upon by UV lights and, PCR inhibitors and whatnot. So we had a lot of gaps and insertions in our DNA sequences. And so, but we got a lot of reads. So we had to collapse them into what we call OTUs, which are operational taxonomic units. These are just used to classify groups of closely related individuals. So we had to collapse them into OTUs. And so we were able to understand what we had, give us a ballpark of what we were working with. And so we aligned those to a reference mitochondrial genome of a dragonfly that was present. We first tried that with Drosophila, and then we were able to find a complete genome of dragonflies in GenBank, and then we're able to extract the alignment to the CO1 region. And so that was what we used for our blasting to try to get our IDs. So we blasted some of our hits, and although this was useful for general IDs, giving us a ballpark to work with, we couldn't get them down to genus and species level. And so we had to use what we call a tree-based method, where we just we combine the sequences we have to reference sequences, and then we're able to build a maximum likelihood tree. And now it gives us a ballpark of gives us an idea of what we're working with, and it gives us IDs to genus level and some to species level. And we're able to, have confident IDs, especially with clades that had, high bootstrap support. And so this just shows the species composition of some of the hits we got. And by hits, I mean number of sequences we recover that were assigned to a given species. And so just for comparative measures, we got samples from Canada, Idaho, and Central Park in New York to compare our results from Nigeria. And we were able to recover a total of 23 hits from Nigerian samples in the tropical region, which was pretty good and promising for future runs, as it also had some overlap with results from traditional sampling, which I had done before as an undergrad. So that was pretty promising. And so this highlights that this, technique is a viable tool for biodiversity assessment in these tropical biomes, although we had some drawbacks, like I said. And so in this, here we were looking at lentic versus lotic sites. And so lentic sites are water bodies that are just still, and lotic sites are flowing water bodies. So we compared the success in lentic sites versus lotic sites, and this study shows the evidence that overall we had, a higher number of recovered odonate species hits in these lentic sites versus than we had in lotic sites. However, that doesn't particularly mean it performed better per se. It just means that in lentic sites we had, it is worth mentioning, in lentic sites we had twice the number of lentic sites than we had lotic sites, excuse me. So we expect that with a level playing field, we'd have, more species hitting lotic sites and lentic sites. And this makes sense because in the lentic sites we had diatoms, molds, and every other thing, drowning out our target organism. So we had a lot of other non-target organisms in our lentic sites, and this just, drowned out our target organisms that we were trying to amplify. And lastly, just to show the potential of this technique, we were able to use this technique to create a haplotype network and just study dragonfly species on a population level. And so we're able to, with one of the species, we got to build a haplotype network and just check for evidence of gene flow in different populations. And it is worth mentioning that on blasting, the sequences we used for this haplotype network came back with over 90% confidence. But on further analysis with the sequences, as we see here, we run into some issues that we'll need to tackle. And although here we see a couple of core haplotypes, we generally see a haplotype network here with multiple nucleotide differences, and there's still a lot we need to figure out, and possibly in the alignment or the entire pipeline itself. And so just a brief background about haplotype networks. It's just a way of visualizing how populations share genetic material. And so these different colors represent the different locations, and the size of the circle represent the population size. And all these tick marks just represent the number of nucleotide differences. So green and pink differ by X number of nucleotides, and that's, in a lot of scientists' opinion, a lot of too much variation. So there's still a lot of debate about how much variation is too much variation. But overall, we see here there's a lot of nucleotide differences, and we suspect that this could just be just stemming from our poor starting DNA quality to or it could just be from the entire pipeline. It could be issues with our alignment. These are things we're still trying to figure out in the entire pipeline. And so just some brief takeaways here. We were able to understand that eDNA is an efficient tool in monitoring and assessing odonate communities. And the success of this technique shows in lotic sites versus in lentic sites, just shows that we have more work to do. And we had things such as molds and diatoms drowning out our target DNA. And so we would need to tweak our primers. So before we start our PCR runs, we have things called primers, which are just what we use to capture the genomic region of interest, and those are the regions that are amplified. So we need to tweak our primers so the primers give us enough matches with dragonflies, but enough mismatches with things such as fish and plants and diatoms, molds, and whatnot. So we had a lot of cases where we had a lot of cases where we had fish amplifying and mold amplifying, diatoms amplifying, diptera amplifying, but we had poor amplification with our dragonflies. So we have we will need to redesign new primers to give us optimum yield for our dragonflies. And so also able to understand that using just blasting for identification is not a good enough way to identify organisms, because the way blast works generally, it just gives you the best first hits, and so it doesn't do a thorough job at searching for the best hit overall or the best match overall. So we try to incorporate that into using the maximum likelihood tree, like I said, where we just concatenate our DNA sequences with with our reference sequences, and we're able to build a maximum likelihood tree, and we just are able to get good species level ID, especially with clades that have high bootstrap support. And so for our future directions, like I said, we're looking right now to, which we are currently working on, just redesigning new primers to increase our success in these lentic sites that previously didn't do so well. So we just get, target regions that are consistent through all dragonflies, but have enough mismatches with these off-target organisms. Sorry. And so lastly, one of the things I'm also looking to incorporate into my thesis is, trying to examine the effect of environmental factors on the eDNA yield, and we're looking at pH, we're looking at temperature, we're looking at biological oxygen demand, we're looking at dissolved oxygen, and how all of these, affect the overall target species DNA yield, and looking at using principal component analysis and structural equation modeling to just see how these factors independently and interactively impact the ultimate species DNA yield. And with that, I'd say thank you for listening, and thank you to my advisor, Dr. Jessica Ware, and Ethan, who helped me with some of these, and everyone in my lab. And with that, I could take any questions.